Now the next field of application is agricultural applications of biotechnology. In other words, something that we also call as green biotechnology. Now green biotechnology, first of all, will be used in a lot of ways. For example, you can use this biotechnology for increasing the nutritional value of a crop, or you can use it to increase the yield of a crop, or you can use it in a manner that the use of pesticides can be lowered. So there are many ways in which we can make the use of biotechnology in the field of agriculture. Now, one core concept to the understanding of agricultural applications is related to transgenic crops. So first of all, we need to understand what is the meaning of transgenic crops, and then after that it becomes easier for you to understand how exactly transgenic crops can be used in various ways in different kinds of applications related to agriculture. So what do we mean by transgenic? So when we use the word transgenic, trans basically would mean different. Trans would mean different. So we are saying that this is a kind of crop which has a different gene. Apart from the normal genes which would be present in that plant, apart from that there are other genes as well. Where is this gene coming from? This gene can come from any organism. Alright, just remember this, that this gene can come from any organism. It can come from another plant, it can come from a microbe, it can come from an animal, it can come from any organism. Just that what we try to understand here is that if there is a property required for a certain crop, we look for genes which have those properties. And if these properties are found somewhere, then we try to see whether or not this gene, wherever or in whichever organism it has been found, whether it can be taken from there and used in this plant. Is it compatible with this plant or not? And in many cases we do find that we are able to make it compatible and we are able to make use of that particular gene. And there are many such examples where we have seen such a use of a transgene. So that's why this kind of a gene which is coming from a completely different organism will be called as a transgene. So that's why these kind of crops will be called as transgenic crops. So that's why these are, in a manner of saying, you can say that these are genetically modified organisms. These are genetically modified organisms. And here, because we are especially talking about crops, we can call them as genetically modified crops. So what happens in a transgenic crop is that there is one or more genes which is coming from not this plant, but coming from some other plant or some other animal or a microbe or something else, meaning that it is coming from some other organism altogether. Then in that case, because that gene has come from some other organism, that's why that gene is called as transgene, and that's why such a crop which has another gene from another organism is called as a transgenic crop. Now why do I need such a gene? Because this gene would have the properties that we needed. Let's say we want to increase the nutritional value of a crop. And to increase this nutritional value, we need another crop or we need a gene from another crop. So we'll take that gene from that other crop and we'll insert it inside this crop and hence try to improve the nutritional value of this particular crop. We want to increase the yield of a particular crop. Then we will look for certain properties which will help in increasing the yield. When we find those properties in a gene in any of the organisms, we take it from there and we insert it inside this plant. So why transgene is required? Because transgene will help in enhancing one property or the other of a given crop plant. So that's why this transgene will be required. So in a transgenic crop plant, one or more genes are artificially inserted instead of the plant acquiring such genes through pollination, while the inserted gene sequence or transgene may come either from another unrelated plant or from a completely different species. Alright? That's why remember that it's not necessary that you are choosing another plant which is very close to this particular plant, but rather you can choose this gene from any species or any organism that you want. So plants containing transgenes are often called as genetically modified crops or transgenic crops or genetically modified organisms. All these terms can be used interchangeably. Now there are certain examples. The most famous of the examples is BT cotton. When we talk about BT cotton is a transgenic crop in which CRY1AC gene of BT, which is a bacteria, is introduced to make the crop pest resistant. So now pest resistant against what? Against bollworm. Now this bollworm used to be a huge menace for the cotton crops. that when we talk about this bollworm feeds on cotton. Alright, it feeds on cotton. It used to eat upon cotton and it used to destroy the crops of cotton altogether. So that's why it was a huge menace. Large amounts of pesticides were used for killing these bollworms or making sure that these bollworms do not destroy the crops of cotton. So this is where biotechnology came to the rescue, that one particular gene called as CRY1AC was taken from a bacteria, a certain bacteria, and this was inserted inside cotton. Now, as soon as this was inserted inside cotton and it became a part of the genetic makeup of cotton, this is where now this variant of cotton, that is BT cotton, became completely resistant to bollworm. Now, bollworm could not feed on cotton because what bollworm needed to feed on cotton was not present in cotton anymore, and this cotton basically became poisonous for bollworm to feed on. So that's why bollworm stopped feeding on cotton altogether. So this slight modification, in terms of introducing this particular gene, made sure that the yield of cotton could increase and we did not need pesticides anymore in these cotton crops against bollworm. So that's why in such a manner what has happened is that we have been able to enhance the property of cotton and make it more usable for us and make it more profitable for the farmers. So this is how you would see that transgenic crops or transgenes would come to the rescue and would help in increasing or enhancing one property or the other of a plant. So why should we make a transgenic crop plant? Basically to make it more useful and productive as far as possible. Now what kind of properties are we talking about? That when we say useful and more productive, what kind of properties? So there are n number of things that you can think of. First and foremost, increasing crop production. How would we increase crop production? There are a couple of ways that you increase the nutritional value of whatever the crops is gaining. How do you increase the crop production? There are a few ways. For example, first of all, you can look at how can you make it flood resistant or drought resistant, so that excess of water or shortage of water does not cause any problem to the crop and crop production still will happen as expected. Second way to do it would be to make it resistant to disease, so that the diseases which would have eaten a lot of crops and would have destroyed a lot of crops, they are not doing that. So one way could be to make it drought resistant or flood resistant. The other way could be that you can make it resistant to the diseases so that crop production does not go down. Another way in which you can think of increasing the productivity is that it should need lesser of nutrients. If it can survive on lesser of nutrients, then in that case you can save some amount there also. And so that's why if you see these lines, transgenic plants have been developed to produce higher yields or tolerate diseases, droughts, etc. Such crops attributes have facilitated increased crop production. So there are two aspects. One, make it resistant to climatic conditions. So resistance to climatic conditions. And second, disease resistance. These are two most important ways to increase the yield of the crops. Then after that, to improve the nutritional value. Now here there is a prime example of golden rice. So when we talk about golden rice, what was done with golden rice is we enhanced the amount of vitamin A in golden rice. Now why did we need to do that? So we know that when it comes to vitamins, vitamins are also called as micronutrients. Why micronutrients? Because they are required in lesser quantities. And many a times the fruits, vegetables, whatever you eat, they have the required quantity of vitamin A and you get your dose of vitamin A from there. But there are areas where people in general would have vitamin A deficiency. So these are the kinds of areas where we need to fortify vitamin A. We need to provide more of vitamin A in these kind of areas. So one of the ways to do that is your food itself should contain the requirement of vitamin A. The required amount of vitamin A should be there in your food itself. This is where a thing like a crop like golden rice came into the picture. Golden rice basically becomes golden mainly because of something called as beta carotene. And this beta carotene is the one that leads to production of vitamin A in the crops. So beta carotene will help in production of vitamin A. Beta carotene is a kind of a gene, and this gene is present in daffodil plant. So now just imagine, on one hand we are talking about rice, on the other hand we are talking about daffodil. two crops which have no relation whatsoever with one another. But still, the genes from daffodil plant could be used in rice, and this beta carotene is the reason why this rice variant becomes kind of yellowish or golden in color, and thus the name golden rice. So if beta carotene was not given, the rice would not be called as golden rice. So this beta carotene contains vitamin A. And when beta carotene is inserted in rice, when it becomes a part of the genetic makeup of rice, this is when golden rice comes into the picture with increased amount of vitamin A, and this is the rice which is administered in the areas which have lower amounts of vitamin A in people in general. So this is how we have seen that increasing of the nutritional value of the crop can also be done. So golden rice is a transgenic variety of rice with genes for synthesis of beta carotene taken from daffodil plant and inserted into rice. Beta carotene is converted into vitamin A. Then after that, it can also be used to increase the shelf life, and especially when we talk about the shelf life of seeds, not only fruits and vegetables, but also of the seeds. Because let's say if we talk about the seeds that have to be used by the farmer in the next sowing season, the farmer has to store the seeds for a very long time, maybe for 6 months to 8 months of time. And in this time period, the seeds may also get destroyed. And there can be n number of external factors. It can be destroyed because of some fungi, because of some bacteria or a viral infection, or maybe because of overexposure to sunlight, overexposure to moisture. There can be many reasons why a seed may get destroyed. So biotechnology can also be helpful in making sure that the seeds can survive for a longer duration of time. So that's why when we talk about increasing the shelf life, it is not only about increasing the shelf life of the fruits and vegetables, but at the same time also increasing the shelf life of seeds. Then after that, environmental benefits. How exactly? This can be done with the help of making crops which do not need pesticides, which are pest resistant in themselves. Because if they are pest resistant, then you will not have to use pesticides on them, and thus you will increase the quality of soil and nearby water bodies. Because when you use a lot of pesticides, a lot of chemical fertilizers, these chemical fertilizers and pesticides will mix with the soil, will seep into the groundwater, will mix with the water bodies nearby, and thus become a part of the entire food chain. So that can become a challenge, because then it can make the entire system poisonous. So if we have crop variants which do not need these pesticides or fertilizers, then we can increase the quality of soil, we can increase the quality of the water bodies also in the nearby areas. So transgenic varieties rely on reduced consumption of pesticides. Consequently, there is less pesticide residue in food, production of pesticides reaching into groundwater, minimization of farm workers' exposure to hazardous products, etc. as well. So that's why basically we are talking about reducing the consumption of pesticides and chemical fertilizers. So what are we basically doing is we are making pest resistant. So that's why you can say that all these are the application areas, or all these are the advantages of making use of a transgenic crop. So if there's a question in the mains examination regarding why do we need transgenic crops, then in that case you can actually use all these points, that why exactly can we, and this question can very easily be asked for 150 words or 200 words or even for 250 words, and depending on what is the length of the answer expected from you, you can also deliberately shorten or lengthen a particular point that we discussed. But these are 4 major reasons that why we focus on making a transgenic crop, because there is another way in which modifications in the genetic makeup can be done, and that is basically through the traditional plant breeding. Now, what is the meaning of traditional plant breeding and how is it different from producing a transgenic crop? Now, traditional plant breeding is when you take 2 very similar species and you try to crossbreed them. You try to cross-pollinate them, and out of that what may happen is that you may get a new variant which has genetics of both these plants, and perhaps you get a better crop variant. So this is the kind of hit and trial that you will do. And there is no scientific methodology here. There is no need of any kind of genetic engineering here. Just that 2 very similar crop plants have been taken together and you are trying to crossbreed them to produce a new variant. This is one way. The second way of looking at traditional plant breeding would be that you selectively breed the plants. What does it mean? Let's say you have 100 seeds of tomato. Alright, and out of these 100 seeds, let's say there are 100 plants that came through. 100 plants germinated from these 100 seeds. Out of these 100 plants, there are 20 plants where the quality of tomato is very good. There are rest 80 where they are either average or maybe not good enough for you. But these 20 are of very good quality. What do you do next? For the next sowing season, you only take the seeds from these 20 plants. And then you use these 20 plant seeds in the next sowing season. So in this manner, what you have done is that you have eliminated the kinds of crops which were either average or below average and not up to the adequate quality that you wanted. But there were 20 which were of very good quality and you selected them. So that's why this is a kind of selective breeding that is done sometimes. So these are some of the traditional methods. Alright, there is no genetic engineering being used, there is no scientific methodology being used, just that you are using your knowledge to your advantage. Whereas when we talk about transgenic crops, we are talking about a transgene, an alien gene which is coming from some other species of a plant or maybe some other organism altogether. And that is being used. So you know what property you wanted and you are looking for this property in all the organisms that you know of. Wherever you get it, you try to extract the gene from there, extract the gene which is responsible for such a property, and then you take it inside the crop plant and thus make the use of that particular gene and make a transgenic crop plant. So that's why these two are different methods. So if you look at the lines given here, traditional plant breeding has been limited to artificially crossing the plants with the same species or with closely related species to bring different genes together through selective or mutation breeding techniques. For example, a gene for protein in soya bean could not be transferred to a completely different crop such as wheat using traditional technique. But in case of golden rice, we have been able to do that. Correct? Transgenic technology enables plant breeders to bring the useful genes together in one plant from a wide range of living sources. So that's why here we are saying that we can actually take any organism. So thus it extends the possibilities beyond the limitations proposed by traditional cross-pollination and selection technique. So traditionally it could either be cross-pollination or it could be selective breeding. Now we are talking about these transgenic crops. One thing that we need to understand is how do we develop these transgenic crops? What is the method behind it? So the method behind this is called as recombinant DNA technology. So we need to understand what is the meaning of this recombinant DNA technology and what do we mean by this term recombinant? How does it produce a transgenic crop? So let's try to understand what exactly happens in a recombinant DNA technology. Now look at this particular figure. Ignore some of the terms that have been used or the technical terms that we have used here. Just focus on the process. Just focus on what's happening here. So first of all, for a transgenic crop, what do you need? You need a gene. A gene that has the property that you wanted. Correct? So where do you get that gene? You might get this gene in another plant or maybe another organism. So you just look for this property. You just look for this gene which would have these properties. As soon as you find them, when you find these properties in a particular gene, you try to isolate that gene first. Alright? So first, the isolation of the gene will happen. So the DNA containing the gene of interest. So let's assume that this is the DNA containing the gene of interest. Now what we do is that we look for a carrier of this gene because the genes will not be accepted directly by the plants. So that's why you need a carrier. Where does this carrier come from? This carrier, which is also called as a vector. This carrier, which is also called as a vector, most of the times will be virus. But sometimes it can be bacteria as well. But in most of the cases, we have seen that we use viruses as the carrier or as the vector. So what we do is that we will take this virus or we'll take this bacteria or we'll take this vector as a general term vector. And what we do is that we try to find a site where we can cut this gene of bacteria or the virus and we can place the segment of the DNA that we needed. Alright. So this is precisely what is happening here. That we have taken a bacteria or let's say a virus, and what we are doing here is that we are trying to recognize the site where we can make the cut. So it is all like a puzzle. Alright. This is like a puzzle where you try to find out the area where you can fit the missing piece of the puzzle. So what we do is that we'll make the cut. Alright. We'll make the cut. So as you can see here, this cut is being made. Alright, this cut is being made. Now as the cut has been made, this is where in this opening the required segment of the DNA can be inserted. So this is what we'll do here, that the required DNA or the required gene sequence will be inserted. Now do you see here that the patterns do match, correct? Here when you cut it open, it is like this, right? And the one which has to be set here, the blue colored portion, this is like this, correct? So this will very easily come here and set with this, right? This will very easily come and set like this, correct? This is precisely what has happened here, that you have made the cuts according to the requirement and you have inserted this required gene here. Now what happens is, at this point of time, there are thousands to lakhs of copies that we produce of this particular recombinant DNA. Alright, so this particular DNA sequence is also called as recombinant DNA. Why recombinant? Because you have cut the portion of the original DNA, inserted it from somewhere else, inserted a foreign DNA here, and then combined it. So that's why it becomes a new kind of a DNA. That's why it is called as recombinant DNA. Now this recombinant DNA, we will have thousands and lakhs of copies of this recombinant DNA. Once we have produced all these copies, now these will be inserted inside the plant. Alright, now it will be inserted inside the plant. As you can see here, this will be inserted inside the plant. So now we have a plant with a new trait. So now this trait or this characteristic, the function, the feature that we wanted in that plant, now this plant has this particular feature. So what have we done? We have first isolated the required DNA from any organism that we wanted or whichever can fit here. We have taken that. We have identified a sequence in the virus or the bacteria where we can set the given gene of our requirement or gene of our interest. Then after that there are thousands or lakhs of copies of it produced, and then after that it will be inserted inside the plant, and then it becomes a part of the genetic makeup of the plant itself, and thus now this plant will have this new feature or this new trait. So this is how the entire process of recombinant DNA technology is carried out. That's why this is called as recombinant DNA technology. And what does recombinant DNA technology do? Recombinant DNA technology produces what? Produces transgenic plants. Alright. So this is the entire process, and thus you have this new property. So all these new properties that we are talking about, whether in case of BT cotton or in case of golden rice, all these basically come via this particular method. The method that you have, the diagram that you see on your screen, via this method only we get this particular kind of new trait or the plant with new traits. So what are the steps? First of all, the isolation of the genetic material will be done, right? The one that we need. So first and the initial step in recombinant DNA technology is to isolate the desired DNA in its pure form, that is free from other macromolecules. Then cutting the gene at the recognition site. Now this is very, very important, because here the role of restriction enzymes come into the picture. What is the meaning of restriction enzymes? Restriction enzymes will basically try to find the area or find the area in this particular gene where the cuts can be made. Alright, that is precisely the role of restriction enzyme, that they determine the location at which the desired gene is inserted into the vector genome. So what is vector? Vector is basically either a virus or a bacteria. So vector is what? Vector is basically the carrier. So these reactions are called as restriction enzyme digestions. So restriction enzyme, what will restriction enzyme do? It will try to find out the location where the cut can be made, where the cut and the insertion of the required DNA can be done. Then after that, amplifying the gene copies through polymerase chain reaction or PCR. So polymerase chain reaction or PCR does what? It will amplify the number of such copies. So this one copy that you had right now thousands to lakhs of copies of this particular sequence will be generated. So there are n number of copies that are generated here. Then after that, ligation of DNA molecules will be done. What is the meaning of ligation? Ligation means nothing but just sticking together. So you have an enzyme which is called as DNA ligase, and DNA ligase is just like a stick. So for example, let's say if you have been using Fevicol, Fevistick and all these things, these are nothing but the adhesives. So when you talk about these adhesives, this DNA ligase also acts like an adhesive. So it's just like a Fevicol or a Fevikwik, where all these cut open portions that you had, they will be stuck together with the help of this DNA ligase. So that's why after restriction enzyme, the other important role is played by DNA ligase. And then finally what happens is that insertion of the recombinant DNA into the host will be done. Correct? So into the host, that is the plant where you wanted these traits to exist, this is the host. So this is where you will insert this recombinant DNA, and after the insertion of recombinant DNA, this genetic sequence may become a part of the plant itself, and thus now this plant will have the new traits or the required traits that we wanted. So this is how the entire process of recombinant DNA technology is carried out. So in this step, recombinant DNA is introduced into the recipient host cell. This process is termed as transformation. This process is called as transformation. Once after the insertion of the recombinant DNA into the host cell, it gets multiplied and is expressed in the form of manufactured protein under optimal conditions. So once this starts to happen, automatically now the genes will keep on getting produced, and thus the plant will have the required property. So this is how the entire recombinant DNA technology is carried out. Look at this particular question. This was asked in your prelims examination. Recombinant DNA technology or genetic engineering allows genes to be transferred. 1. Across different species of plants. 2. From animals to plants. 3. From microorganisms to higher organisms. Select the correct answer using the codes given below. 1 only, 2 and 3 only, 1 and 3 only, and 1, 2 and 3. Simple question. I think this is something that I have been repeating over last 10-15 minutes, that gene can come from anywhere. So across different species of plants, yes, it is possible. From animals to plants, yes, this is possible. From microorganisms to higher organisms, yes, this is also possible. So that's why the correct answer here will be 1, 2 and 3. As I said, that it can be any organism. It can be any organism, because if it was just about different plant variants, then it was a regular or a traditional way of breeding plants. But in this case, it can come from any organism. Now the next agricultural application of biotechnology is through biofuels. Now in the chapter of energy, we have discussed about biofuels, and there we have discussed how biofuels can be produced via different food crops and non-food crops, agricultural crops as well. So in this case, that's why biofuels can also be called as a kind of green biotechnology, because you are making use of some of the agricultural crops in the production of biofuels. For example, we had talked about corn, we had talked about soya bean, we had talked about oil seeds, and all these can be used for the production of biofuels. So that's why biofuels also can be seen as one of the agricultural application of biotechnology. Now, so that's why one of the biggest applications of biotechnology is in the energy production sector, with fears over dwindling oil resources in the world and their related environmental impact. There is a need to protect the globe's future by finding alternative environmentally friendly fuel sources. And this is something that has been made possible with the help of biotechnology. And why are we calling this as biotechnology? Reason is simple. You are using a crop. You are using something which is living or recently living. So that's why biofuels also, when you are using agricultural crops, biofuels also can be called as a part of green biotechnology or agricultural application of biotechnology. Now the next application area is biofortification. Now what is the meaning of fortification first of all? Fortification is adding of some ingredients or some nutrients to a given crop. Like for example, we discussed golden rice. Now in case of golden rice, what were we doing? We have added vitamin A to golden rice, or we have added vitamin A to rice which has become golden rice. So in this case, now this golden rice will be given to the areas or will be supplied to the areas where there is a deficiency of vitamin A. And there are cases of diseases which are caused due to deficiency of vitamin A. So that's why this is how biofortification works. That with the help of biotechnology, you will add certain nutrients, you will add certain ingredients, components to the crop, making sure that this crop now has a little more amount of some kind of nutrient, and then it can be supplied to the areas which are deficient in that particular nutrient. And this is something which is done mainly for the micronutrients. Micronutrients are the nutrients which are required in smaller amounts, like vitamins and minerals. They are called as micronutrients. So for that, we generally can use the process of biofortification. So the development of micronutrient dense staple crop varieties using the best traditional breeding practices and modern biotechnology can be called as biofortification. So that's why it comes from bio and fortification. Fortification would mean the addition of the ingredient to the food, and bio because we are doing it with the help of biotechnological tools. So that's why it becomes biofortification. Now, apart from biofortification, another way to take care of this kind of a problem is through supplementation. What is the meaning of supplementation? If you are taking the supplements of these nutrients, that many a times you might have seen that you might take vitamin B12 capsules, vitamin C capsules, vitamin D capsules. So all these are nothing but supplements, that you are not taking the vitamins through the food, but rather you are taking these vitamins through medicines. So that's why it is a supplementation of what is required. Alright. So biofortification and supplementation, these two work hand in hand. If biofortification can work, good. If not, then supplementation is the way to go. Then after that, there is something which needs to be addressed today, and this is the issue of hidden hunger. What is the meaning of hidden hunger? Now, let's say you are talking about a poor nation where the supply of food is not adequate. So what is being done by many of the other countries is that these countries are providing the food supplies to these countries, these poor countries. Now, the food supplies may be enough, but many a times the nutritional value may not be enough. Alright? Food is enough, that your stomach is getting filled. You are getting enough of food, but this food does not have enough of all the nutrients which are required. So they may have higher amounts of proteins and carbohydrates, but they may not have sufficient amount of vitamins and minerals. So in this case, what happens is that one component or the other of the nutrients that are required is missing from your diet. And this is the case which is called as hidden hunger. That although you have eaten enough, you are getting enough of food, but you are not getting enough of nutrients. Alright? You are getting enough food, but not enough nutrients. Alright? Enough food, but not enough nutrients, and especially these would be micronutrients. So in these cases also biofortification will be required, so that we are able to the non-existent part or the limited part of the nutrition in the food. So this is something that has started to become a problem in the world, that the issue of hidden hunger has been seen as a problem, especially for the poor nations, where although supply of food is adequate, but it is not a balanced supply of nutrients. So that's why this is a problem which is called as hidden hunger. Then after that, another way in which application can be seen is pest and herbicide resistant cultivars. What is the meaning of cultivars? Cultivars are basically the crops which are being grown. So we had taken the example of BT cotton. So BT cotton is an example where we have created a cotton variant which is resistant to a pest called as bollworm, and it is able to protect itself against bollworm without any external help of any pesticide, etc. So that's why when we talk about pest and herbicide resistant cultivars, what happens in these kind of crops is that they are able to themselves, by their mechanism, they are able to resist the pests, and they do not need anything externally. Then after that, you have herbicide tolerant crops. So herbicide tolerant crops are the ones which will not be affected by the herbicides. Now what happens is, let's say in a given crop field, what may happen is that apart from the crops that you are growing, there are many weeds that have grown in the same field. So in this case, you would need herbicides so that you can kill these weeds. But at the same time, you also want that your crop should not be harmed, correct? You do not want your crop to be harmed. So in this case, we need herbicide tolerant crops which will not be affected by the herbicides at all, so that whenever you spread these herbicides, it will kill all the unwanted weeds, but it will have no effect on the crops. So that's why these kind of crops are being also developed. Then after that, in animal nutrition. Now when you talk about animal nutrition, in what kind of ways can it help in animal nutrition? First of all, we know that the cattle or the livestock also need to have enough of nutrients so that they can perform all the tasks that we want them to perform, whether it is related to agriculture or whether it is related to dairy or anything. So for that, they need enough of nutrients. Now the challenge for the farmers many a times is that to provide enough of nutrients to the cattle and the livestock, they have to spend a lot of money. So that's why many a times we try to make food or the animal food in a manner that although they are being consumed in lower amounts, but they have enough of nutrients that were required by the cattle or the livestock. So in this case, the farmer does not have to spend a lot in ensuring the food and nutrition for the livestock. So this is also one way of doing it. So biotechnology has already resulted in animal nutrition aids such as enzymes, probiotics, single cell proteins, and antibiotic feed additives that are already widely used in intensive production systems worldwide to improve the availability of nutrients from feeds and the productivity of the livestock and aquaculture. So what do we want? We want less food, more nutrition. For our animals, what we need is less of food and more of nutrition, so that even in a limited supply of food, they can get enough of the nutrition that is required for their daily activity. Then the next is use of biofertilizer. Now again, use of biofertilizer is always a much better alternative as compared to chemical fertilizers, because in most of the cases the chemical fertilizers are poisonous, and once they become a part of the food chain, maybe through the crops, maybe through the soil, maybe through the nearby water bodies, then in that case they can lead to the problem of bioaccumulation, and ultimately it can become poisonous for the animals who are living in the water bodies or even for the humans via the food chain. At the same time, they are not very good for soil health as well. For soil health, excessive use of chemical fertilizers, it leaches the soil of the nutrients, it makes the soil devoid of the required nutrients, and that's why it is not really good for the soil in a long run. But as compared to that, when we talk about biofertilizers, meaning the fertilizers which are coming from biological origin, these are fertilizers which are much better alternative for maintaining soil health and also for the environment altogether. So that's why use of biofertilizers is another application area in agricultural application of biotechnology. Then improvement in floriculture. Now what do you need out of floriculture? What do you need out of flowers? You need them to have good colors, correct? Then their scent matters, their size would matter, their lifespan would matter, correct? So through the use of biotechnology, we want to enhance these properties, that they should be colorful, they should show different kinds of colors which may be attractive, they should have a good scent. At the same time, they can have a good size which also again can look very very beautiful, and they should have a good lifespan. It shouldn't happen that the flower grows today and dies tomorrow. You would want it to be there for maybe 15 days, 30 days of time, so that whatever you have spent on floriculture, you are able to get out of it. So that's why these are some of the characteristics or features that you would want in flowers. So this is what can happen with the help of biotechnology. This is another way in which biotechnology can help in the field of agriculture. So floriculture is associated with flower cultivation industry. Biotechnology is playing key role in generation of new variants, new varieties as improve. So floriculture is associated with flower cultivation industry. Biotechnology is playing key role in generation of new varieties as improvements in color, scent, size and flower long life through gene manipulation techniques. So this is what we want, that we want these kind of properties, and we would make changes in the genetic makeup of the flowers so that these kind of properties can be enhanced in the flowers. Look at this question. This was asked in your prelims examination. Other than resistance to pests, what are the prospects for which genetically engineered plants have been created? One, to enable them to withstand drought. This is something that can be done, and we have discussed about this. To increase the nutritive value of the produce, again biofortification, we discussed about this. To enable them to grow and do photosynthesis in spaceships and space stations. Now let's hold on to this. This is a very interesting statement. To increase their shelf life. Again, this is something that we have discussed. So 1, 2 and 4 are definitely correct. Alright. So C looks to be correct, but D also can be correct. A and B definitely are not correct. Right. Coming back to statement number 3. Remember that this is a 2011 question. To enable them to grow and do photosynthesis in spaceships and space stations. So what are the prospects for which genetically engineered plants have been created? Alright. The question is about have been created, not can be created. If it was about can be created, definitely 1, 2, 3 and 4 were correct. But since the question had asked have been created 2011, this was wrong. And that's why the correct answer here is 1, 2 and 4 only. If instead of have, if this was can be created, then in that case 1, 2, 3 and 4 would have become correct. Now, if you talk about statement number 3 in today's context, you will find that there are some experiments which have been done by NASA back in 2015, 16, etc., where they have actually been able to grow plants in space. So maybe in the present context, if the same question comes back, then in that case the correct answer could be 1, 2, 3 and 4. But talking from the perspective of 2011, the correct answer, the official answer given by the UPSC at that point was 1, 2 and 4. So that's why this becomes a very interesting question. In today's context, perhaps we can say 1, 2, 3 and 4 are correct.